When I was a child, I loved eating corns. I had noticed that some corns were yellow colored, some were dark purple colored, and some were even mosaic colored. I was also a curious child that always wondered why the corns had so many different colors. I didn't understand this mystery until I went to university and studied biology and genetics. So, why corns are yellow? Every corn kernel has multiple layers. The yellow color of the kernel is the color of the endosperm. If the outer lower layer of corn is transparent without anthocyanins, it will show the yellow color of the inner endosperm. Then you may ask, why some corns are mosaic colored? Well, the American cytogenetist Barbara McClintock wondered the same thing. Barbara McClintock identified two new dominant genetic loci. She named these two loci dissociation and activator transposable elements. Transposition of dissociation, activator, or both can result in chromosome breaks or fusion. However, transposition of the dissociation occurs only when the activator is present in the nucleus. She found that the purple color in maize is regulated by this dissociation activator two-unit system. They can cause the expression or suppression of anthocyanin pigment genes, such as colored alarm gene and bronze gene. Because the transposon jumps very actively and fast in chromosomes, the expression of anthocyanin starts and stops from time to time. The expression level of anthocyanin in different kernels will also be different, and hence the purple pigment will sometimes be darker, lighter, and even spotted. Apart from the colored alarm gene and the bronze gene, many other loci are also responsible for the synthesis of the purple anthocyanin pigment in the alarm layer of the kernel. For example, anthocyanin gene 1 and anthocyanin gene 2. In this experiment, these two selected loci along with the shrunken gene and waxy gene were focused to investigate the mutability of anthocyanin gene 1 and anthocyanin gene 2. Mutations occurred in anthocyanin gene 1 and anthocyanin gene 2 are designated as follows. When the kernel has a homozygous recessive allele, anthocyanin-less gene 1 or anthocyanin-less gene 2, there will be no anthocyanin pigment formed in the alarm layer, so the kernel will be colorless. Mutations in these loci would result in different colored kernels. Now we want to know whether these mutations are activator-controlled. McClintock constructed activator test stocks homozygous for inhibitor gene, shrunken gene 1, bronze gene, waxy gene, and dissociation transposon in chromosome 9. She crossed 29 plants from culture 6424 with the activator test stocks. These activator test stocks had genotype of homozygous inhibitor gene, dissociation gene, and anthocyanin gene 1. Results show that group 1 plants, which had no activator transposon, had all colorless resulting kernels. Group 2 and Group 3 plants, which had one or more activator transposon, had both colorless and variegated resulting kernels. Then she crossed these 29 plants with tester plants that had homozygous anthocyanin-less gene 1 and shrunken gene 2, that with and without the presence of an activator. Results show that colorless resulting kernels were from Group 1 and variegated kernels were from Group 2 and 3. And that makes sense. From the above experiment, she could determine that mutation at anthocyanin-less genes are controlled by the activator transposon due to the induced chromosome breakage or dissociation insertion. This experiment was a remarkable achievement that predated the discovery of regulatory genes in eukaryotes by several decades. Barbara McClintock won the Nobel Prize in Physiology or Medicine for this research. The most outstanding thing about Barbara McClintock is that she completed such magnificent scientific research on her own, with the most unflinching confidence.